Our last speaker for the morning session is Dr. Rick Lindroth. Rick received his BS in wildlife biology at Iowa State University and PhD in ecology at the University of Illinois. He joined the faculty at the University of Wisconsin in 1988 as professor of ecology in the Department of Entomology. Rick's research interests address chemical mediation of ecological interactions and range from plant biochemistry to ecosystem function. His primary focus is on the chemical ecology and evolution of aspen herbivore interactions. He collaborates on the functional genomics of phenolic production in aspen and hybrid poplar. Rick has over 140 publications in peer-reviewed journals. He's a 2009 recipient of the Silverstein-Simeon Award from the International Society of Chemical Ecology for his work in the chemical ecology of aspen. Rick?
Well, thanks to the organizers for the invitation to speak. I'm always struck as someone from the upper Midwest coming to the West by the magnitude of problems that are being experienced with aspen here. Back in the upper Midwest, it grows vigorously. We don't have many of the problems that you experience here. So in this last lecture this morning, I want to just step back from the landscape level for a few moments and talk about things at a biochemical or physiological level, and then we'll work our way back out to the landscape level by the end of the talk. I don't need to go over most of this kind of background information that I give in most lectures about aspen. You're all aware that it's a dominant species in many habitats. It has extraordinary genetic variation as well as terrific phenotypic plasticity, extensive spatial and temporal distribution, and for particular interest in this talk, it serves as a host to well over 100 different species of herbivores, probably 200 species at least in North America. So what I'll talk about this morning is outlined here. First of all, the basic phytochemistry of aspen. Spend a fair amount of time talking about the factors that influence phytochemical variation, including genetic, environmental, and gene by environment interactions, developmental variation, and then talk briefly about costs of defense. And then we'll look at the effects of phytochemical variation on herbivores as well as ecosystem dynamics. All right, so phytochemistry. Those of you who have no interest in biochemistry, you can just close your eyes and snooze for about 1 minute here. Yesterday, Sam talked about the importance of carbohydrates as resource pools for either growth or defense. If they're used as defense agents in poplar, they go through this pathway, the shikimic acid pathway, leading to, of interest to us, a suite of compounds known as phenolic glycosides, or salicylates, and condensed tannins. So these are the dominant phenolic-based defensive compounds that occur in populace. A little bit more about the structures of these. The phenolic glycosides or salicylates have this type of structure. This is glucose, so these are glycosidic compounds. This component right here is salicyl alcohol, structurally very similar to acetylsalicylic acid, which we know as aspirin, and various additional function groups that are tacked onto these. Aspen produces 4 different salicylate compounds, 2 of them in fairly high concentrations, tremulacin and salicortin. When I talk today, we typically just pool those together. Because tremulacin and salicortin are structurally fairly similar, I'll just be talking about phenolic glycosides or salicylates as a composite pool. In addition, aspen produces fairly sizable concentrations of condensed tannins, which are these very large polymeric compounds structured with basic flavonoid units. Okay, that's the chemistry lesson for today. There are many different pharmacological effects of aspirin salicylates known in the human medical literature. These compounds are analgesic, anti-inflammatory, antipyretic, that is, they reduce fevers. They inhibit blood platelet aggregation, alter uric acid excretion, dissolve warts and corns, inhibit bone metabolism, inhibit proteoglycan synthesis, and can contribute to peptic ulcers. take-home message is they have many different types of modes of action. They attack many different key points in the physiology of animals. Which leads us then to how they might function with respect to herbivores. First of all, they have fairly low potency. It takes large amounts of these compounds to have much of an economic or much of a physiological impact on herbivores that are adapted to feeding on aspen. So they also have low specificity, that is, they're broad spectrum. They affect fungi, they affect insects, they affect mammals, they affect birds. And they occur at relatively high concentrations, 1 up to 20, sometimes 25% dry weight of a leaf can be comprised of these phenolic-based constituents, so whopping concentrations. In fact, this gypsy moth caterpillar feeding on an aspen leaf with modest concentrations of these salicylate compounds, if you were to translate its consumption of salicylates into human terms, it would be like a human eating about 7 pounds of aspirin in a day. So that's the translation to what these guys are processing in terms of salicylates. Of course, they have metabolic adaptations that allow them to process these things fairly efficiently, but not without some cost or damage to themselves. In fact, at high concentrations, these compounds are deterrent, they are toxic to even gypsy moth or aspen adapted insects such as gypsy moth. They cause massive lesions in the gut and septicemia. And these things attack multiple target sites. So let's take a look now at phytochemical variation, and we'll focus first on genetic, environmental, and G by E factors. And the take-home message of the next 2 slides is just to impress upon you the tremendous genetic variation in western aspen with respect to production of both phenolic glycosides and condensed tannins. So these are 31 different clones randomly selected and sampled in north-central Colorado. We've done the same in central Utah and the same now in northern Arizona, and the results are all the same. Tremendous genetic or clonal diversity in production levels of these compounds. Each of these clones was sampled 4 to 5 times, 4 to 5 different rabbits. You can see that the amount of variation within a clone is actually quite small. The amount of variation among clones is really very large. So phenolic glycosides can range from 5 up to 25% of the dry weight. Interestingly, and I'll come back to this, if we were to compare a similar chart from Great Lakes aspen to these western aspen, all of the composite phenolic glycoside concentrations would peak around 5 to 6% in the Great Lakes. So levels of phenolic glycosides in adult mature aspen in the Great Lakes region is much lower than it is here. And so far we don't have a good explanation for that. Levels of condensed tannins are also very highly variable among clones, also in Colorado, Utah, and Arizona. So one of the questions that we started addressing a number of years ago in my research group is, what is the cause of this tremendous variation? Is it genetic, is it environmental, or is it a consequence of interactions between genes and the environment? Is it nature, is it nurture, or is it the particular nature in which you're nurtured in? So we looked at a variety of factors, including light, this is a shade house, soil fertility, drought, and other factors to see which of these factors are driving expression of phenolic glycoside and condensed tannin traits in aspen. These two geneticists are known as norm of reaction plots. What does that mean? Each of these lines represents a different aspen clone. And these are clones that were grown either under low light or high light, or under low soil fertility or high soil fertility. These are the results for phenolic glycosides, and as you can see, most of the variation in this figure is clonal. That is, the variation here or the variation here. Yes, there is a slight effect of light. This group is a bit higher than this group, so high light does tend to enhance phenolic glycoside production. There's a very modest effect of soil fertility, and in fact, it tends for some to go up and others to go down. So soil fertility is kind of a washout in terms of production of phenolic glycosides. The take-home messages, these are driven primarily by genetics. In fact, they're almost genetically canalized. That is, there's very little response to environment in terms of expression of phenolic glycosides. It's largely genetically clonally based. Now, compare this for the phenolic glycosides to the condensed tannins shown here. This is a great example of a very strong gene by environment interaction. That is, we have strong expression of the genetic component, but that expression of genetics is determined by the environment in which they're grown. If they're grown under high light, we see strong variance among different genotypes. If they're grown in low light, there is hardly any variance among the genotypes. So in this case, we see both strong genetic, environmental, and G by E effects affecting condensed tannins. What about induction? We've done a number of different studies looking at induced responses. If you have insects feed on aspen, we will see modest induction, very slight induction of phenolic glycosides, stronger induction of condensed tannins. If you feed them extensively and you come back a year later and measure foliage on plants a year later, you'll still see the signal of strong induction in condensed tannins, very little, if any, induction in phenolic glycosides. The only cases in which we've seen phenolic glycoside production increase is if you completely eliminate the leaves and you get a reflush. And that's very similar to what Sam's research showed, he reported on yesterday. If you have complete frost damage and a reflush of leaves, you get higher levels of phenolic glycosides. But in the leaf remnants themselves, they don't seem to induce to any great degree. All right, see I didn't correct one title there, but there is also developmental variation that we've observed in expression of secondary chemistry. If you do a transect across a clone and measure levels of phenolic glycosides and tannins expressed in different age classes, this again is for Wisconsin clones. There's a strong marked decline in expression of phenolic glycosides. Each line represents a different clone, and an increase in expression of tannins. So adult trees in Wisconsin have relatively low levels of phenolic glycosides, high levels of tannins. How does this compare to Western aspen? A very similar response, except that they don't drop nearly as low. So they would tail off about in this region here in Utah or Colorado. Does expression of chemical defense exact a cost? One would expect that if these things really are defensive, that there would be strong evolution to select for clones that all express high levels of phenolic glycosides as defense. Why is it that during outbreaks of, let's say, the forest tent caterpillar, you see some trees that are completely or nearly completely defoliated and others that are not? Well, the conventional view is that these things are probably costly, so that in years where you don't have high exposure to herbivores, it's costing the plant in terms of fitness to express high levels. And so you have this basically polymorphism in the population. You have some genotypes that might produce high levels and grow slowly, or produce low levels and grow well. Is there any evidence of that? Well, yes, indeed, there is. Again, based on a number of potted studies in common gardens that we've done with our aspen. I think I've had some problems with shifting to the newer PowerPoint, because I've got several slides that are popping up incorrectly. Anyway, bear with me. Costs of defense are indeed environmentally mediated. What we see here is each dot represents a different aspen clone. And these clones were grown under conditions of low light, low fertility, low light, high fertility, high light, low fertility, and high light, high fertility. The take-home message is there's a strong negative genetic correlation between final tree weight and levels of phenolic glycosides. Those trees or those clones that produce high levels of defenses grow slowly. Those that produce low levels of defenses grow well. And that pattern holds true in all but one case, and that is when you have a surplus of resources. So trees grown under conditions of high light and high fertility, there is not this strong negative genetic correlation. kind of like the federal government. If the economy is booming, you can have both strong military and strong social programs at the same time. But as soon as resources are limiting, then you've got a choice to make. Are you going to put your resources into defense? You're going to put it into growth. And so that is, in each of these cases where resources are limiting, there's a strong negative correlation between tree growth and defense traits. So to summarize, in terms of response of chemistry to various genetic, environmental, and developmental constraints, we see that phenolic glycosides are strongly influenced by genetic factors, weakly influenced by resource availability, weakly influenced by defoliation, with the one exception of if you completely eliminate all of these on a tree, and strongly influenced by developmental stage. Condensed tannins are also strongly influenced by genetics and by resource availability, modestly affected by defoliation, and also strongly affected by development. Okay, well, what you're all waiting for, what does this really mean? What are the implications for defense of aspen against herbivores, and what might be the implications at the ecosystem level? We've done a number of studies, and there are also a couple of other studies in the literature which have shown that phenolic glycosides reduce either preference or performance of a number of different herbivorous species. So we've documented that they reduce performance of gypsy moths, they reduce feeding, they reduce survival, they reduce growth rates. Same thing for forest tent caterpillars, same for large aspen tortrix, white-marked tussock moths, tiger swallowtails, and big poplar sphinx moths. Keep in mind that all of these are aspen-feeding insects. All of them are adapted to feeding on aspen, but they're not adapted to feeding on all aspen equally. So those aspen that are well defended will indeed reduce the performance of these numerous insects. Some of these are actually specialists, like the big poplar sphinx moth. It doesn't really eat anything other than poplar, but even its performance is dramatically reduced when feeding on well-defended clones. There are a number of studies that are starting to come out now with mammals. We know that mountain hare, porcupines, and elk are also influenced by phenolic glycosides. I'll show you some of those data, as well as some fungal pathogens are influenced by phenolic glycosides. To date, there's very little evidence suggesting that condensed tannins have any impact on herbivores whatsoever. We've done a couple of studies indicating that tannins are probably influential in modifying consumption by leaf-feeding beetles. They don't seem to affect most herbivores and most lepidopteran feeders, and they don't seem to have much of an effect on mammals. So I'm going to be really focusing on the phenolic glycosides. We can talk about the condensed tannins afterwards if you're interested. Here's one example. These are gypsy moth caterpillars of the same age, reared on one of 7 different clones of aspen. They're reared to pupation. What this shows is female pupal weight. There's a threefold variation in female pupal weight, which 98% of that variation is explained by the levels of phenolic glycosides that occur in their host plants. So a very strong correlation between the performance of the insect and levels of phenolic glycosides. And we've seen the same thing with forest tent caterpillars with different indices of performance, such as survivorship, feeding, etc. Work by Jack Donaldson, a recent graduate student, showed that indeed, in an experimental sense, if you apply gypsy moths to an experimental common garden of aspen trees of different genotypes, we indeed do find changes in defoliation rates, and levels of phenolic glycosides are inversely correlated with defoliation. So the better defended trees receive less defoliation. But key point, phenolic glycosides are ineffective during peak outbreaks. This is a shot taken in midsummer in northern Wisconsin in about 2001 or 2002 during our most recent forest tent caterpillar outbreak. These insects go through outbreak cycles. Every 8 to 12 years there's another outbreak. In 2001, 2002, there were close to 7 million acres of land in northern Wisconsin and Minnesota and northern Michigan that were completely defoliated. When these population densities get that high, they're feeding on ferns, they're feeding on conifers, they're feeding on all kinds of things that they would normally feed on, and the aspen is completely defoliated independent of its level of defense. What about browsing by mammalian herbivores? What evidence do we have that these compounds are defensive? As I mentioned, a couple of studies are now emerging, some with porcupine, elk, and we're starting to do some work with deer. This is a study done by Stuart Woolley in southern Utah a couple of years ago. We worked with a rancher who had some captive elk. We collected aspen from a number of different genotypes and fed it to captive elk. And as we had anticipated, there is an inverse correlation between levels of phenolic glycosides and elk consumption. So these compounds do tend to deter feeding by elk. And in fact, we've seen already a couple of slides like this the last couple of days. This is a shot from Rocky Mountain National Park. This exclosure was put up a number of years ago when the aspen trees were quite small. Those outside the exclosure continue to get hammered. Those inside the exclosure, of course, grow quite well. This is a shot outside the exclosure. And when browsed by elk, these little shoots, saplings, revert back to kind of a juvenile growth form with high levels of phenolic glycosides and low levels of tannins. So here's an example of two different clones that we measured inside and outside of exclosures. The mature trees had, in both cases, lower levels of phenolic glycosides than do the immature resprouts. So in terms of response to browsing, these trees do kind of revert back to a juvenile form with expression of high levels of phenolic glycosides. And similarly, as we would expect based on our data from development, lower levels of condensed tannins. So again, browsing tends to revert the trees back to a juvenile growth form, high levels of phenolic glycosides, low levels of tannins. This is work done by Joe Bailey and several other colleagues at Northern Arizona University. We looked at browsing by elk and how it reconfigured the population of aspen following fire and resprouting of a number of aspen shoots, and then they were protected from elk browsing for a period of some years. Then the elk came in and they selectively browsed out certain genotypes. And what that did in this logistic regression is it shifted the remaining genotypes toward higher levels of expression of tremulation, one of our phenolic glycosides. So elk apparently do exert a selective force in shifting the defense expression of aspen populations. And more recent work by my group in Wisconsin has shown that deer also browse selectively on aspen genotypes. These are two different genotypes of aspen exposed to deer browsing. One is not touched, the other, almost all of the leaves and shoots have been removed. So considerable variation among different genotypes. We don't yet know what the basis for this selective feeding is, or whether it's phenolic glycosides or other compounds. We're working on that. So the obvious question then is, why does phytochemistry not protect aspen against excessive browsing in the Intermountain West? You're all familiar with scenes like this where these aspens continually get browsed year after year. And I think the answer to the situation here is similar to the answer in the Great Lakes region when we have outbreaks of forest caterpillars. And that is simply that even poor food is better than none. If you're forced to eat bad aspen or conifers, well, you're going to eat bad aspen. All right, but if the population densities, I would wager that if population densities of elk or cattle were low enough, that the defense characteristics of the trees would express themselves in differential browsing. In fact, several of you have mentioned that in the field you can sometimes witness even clones side by side where there is better regeneration under some clones than under others. So let's talk for the last few minutes about effects of chemistry on ecosystem dynamics. Aspen chemistry can influence ecosystem function directly or indirectly. It can do so directly via leaf litter fall, decomposition, root exudates entering the soil, or aspen chemistry can influence ecosystem function indirectly by changing rates of frass fall, fecal fall, and green leaf fall. I'll just focus on some of the direct functions here for a few minutes. This is a study done by Mike Madrich in my group, published just, I think, a year or so ago in Ecosystems, where we looked at decomposition in 5 different clones of aspen grown under conditions of low or high soil fertility. And what we see here is the percent of leaf litter remaining over the course of about a year. And basically the message is the clone, as well as the condition in which those clones are grown in, will considerably influence rates of leaf litter decomposition. And this differential decomposition was strongly linked to chemical variables in the plants, especially levels of tannins. So here's where the tannin story starts coming into play. Phenolic glycosides really don't affect decomposition rates. Tannins do, lignin does, nitrogen levels obviously do as well. Mike has gone on to look at landscape variation in ecosystem function. This is an aerial photograph of an area that Mike works in just north of Madison, Wisconsin. Each of these little clusters is a separate aspen clone, and microsatellite DNA analysis indicates that these are indeed genetically different clones. And what he's done is he's conducted reciprocal transplants with moving litter from one clone to another. He's looking at soil microbial activity. And at the landscape level, we're seeing that the genetic composition of the clone will influence microbial characteristics ecosystem characteristics such as litter decomposition, and soil microbial characteristics such as extracellular enzyme activity and even soil nitrogen cycling. So at the landscape level then, we're seeing this mosaic of ecosystem function that is being driven primarily by the genetics of the plant. And this is similar to work that we've been involved with colleagues at Northern Arizona University, Tom Whittam and his group, where in riparian systems throughout much of the Mountain West, and they've documented this in several systems. I think their major work has been done on the Weaver River just south of here. At the low elevations, the riparian habitats are dominated by Populus fremontii, Fremont poplar. Then there are these extensive hybrid zones where Fremontii hybridizes with Angustifolia, or narrow-leaf cottonwood. Well, Fremontii has low levels of foliar tannins. These decompose quite rapidly at the lower elevations, and you have high nitrogen mineralization going on in the soils. At the upper elevations, Angustifolia produces high levels of foliar tannins. These compounds slow or decrease decomposition rates, and you therefore have low nitrogen mineralization at these higher elevations. And in the hybrid zone in between, you have intermediate levels of these processes. So again, the take-home point here is that the plant genetics are strongly influencing the plant chemistry, which in turn influences ecosystem function. I'm sorry about all my titles being funky. The genetic variation then and extensive clonal growth of aspen likely result in spatial mosaics of ecosystem function at the landscape level. And both Mike's work and the Northern Arizona group's work is starting to show that in a number of different habitats. Finally, just some comments about some work that I hope to be involved in Yellowstone National Park with some colleagues both out west and in Madison. You're all aware of the extensive fires in Yellowstone in 1988, leading to a flush of actual sexual reproduction seedlings, which then are being selected for either by browsers or via competition with other aspen or competition with lodgepole pine. And these interacting biotic as well as abiotic factors will eventually lead to differential expression of these genotypes across the landscape level. So what we hope to do is actually assess the magnitude of selection imposed by both browsing mammals, as well as competition, as well as some climatic factors, and see how that alters the landscape genetics of aspen at these large spatial scales. So to sort of wrap up how all these things come together, and don't get too excited, I'm not quite finished because I threw in a couple of slides at the end as a bonus, so bear with me. But what we see are genetic variation and environmental variation, both influencing plant photosynthesis, growth and reproduction, and altering the chemistry of aspen. Altered chemistry in turn will change rates of herbivory, susceptibility to disease, differential frass or fecal quality. These will alter nutrient cycling and feed back to affect the environment. Changes at this level can affect stand structure and variation in the herbivore community that's associated with aspen, which in turn can feed back over evolutionary time to change the genetic variation. And genetic variation and environmental variation together operate to influence variation at the landscape level. So this is kind of just putting all the various pieces together. These are the bonus slides. Several of you have mentioned aspen in the context of climate change. I've also been involved for about 10 years with a group working at the Aspen Face Site in northern Wisconsin. This is one of about half dozen free air CO2 enrichment or face sites in North America. This is the largest one in the world. We have 12 face rings. We're monitoring or we're manipulating both CO2 and ozone. So this is a complete factorial. We have 3 replicates of control, 3 of high CO2, 3 of high ozone, and 3 of high CO2 plus ozone. CO2 is regulated at about 560 parts per million, and ozone is 1.5 times ambient, or about what you would expect in Milwaukee, Chicago on an average summer day, less unsure than what you would get in Salt Lake City on an average summer day. And we're fumigating stands of aspen, birch, and maple. This has been going on for about 12 years. During the summer, we're pumping about $6,000 worth of CO2 into these rings every day. So it's a fairly sizable and expensive project. And I just want to show one slide because this documents that aspen can be an effective sequesterer of carbon, but that the efficacy in carbon sequestration is going to be influenced by other factors such as ozone pollution. In the black, which is pretty much covered up by the red, is the growth of aspen trees. They started out as seedlings in the control rings. Blue is the elevated CO2. So we see enhanced growth under high levels of CO2, and that is not showing any sign of tapering off, as people predicted would happen some years ago. So CO2 definitely does enhance growth of these trees. Ozone diminishes the growth of the trees, and when you have ozone and CO2 coupled together, they're growing at about what ambient level CO2 trees are growing now. The take-home message is that, yes, these trees do have the capacity to sequester more carbon in the future, but that is going to be inhibited by ozone. In fact, in this case, it's completely offset by ozone pollution, which itself is becoming more and more of a regional problem. And finally, one other feature of aspen in relation to climate change and CO2 is documented here. This is a rather complex-looking graph. Actually, it's really slick, so just bear with me as I talk through it. This is work done by Chris Cole, who did tree ring analysis of about 900 different trees from aspen clones scattered throughout Wisconsin. He then ran it through a very sophisticated statistical analysis relating tree growth to actual changes in CO2 concentrations in Wisconsin over the last 70 years, to the Palmer Drought Severity Index. So on this axis, these are droughted trees or drought years. Up here, these are very moist years. And then related them to average diameter growth in these trees. What we found is that growth of natural stands of aspen has already increased due to the increased levels of CO2 in the atmosphere. In fact, they've increased by 39%. So already the very modest effect, the very modest increase we've already experienced in levels of CO2 is having a fairly pronounced effect on growth of aspen in Wisconsin. If we look at, let's look at this curve right here. This is under very dry conditions. What we see is that as CO2 increases, the growth of the trees increases, but only marginally when it's very dry. If we compare that to this curve, what we see is under wet years, the response to CO2 is very pronounced. So as the environment gets wetter, you have a strong response to CO2. As the environment gets drier, there's a weaker response to CO2. But across the board, there's an increase of, actually this dimension, across the board there's an increase in tree growth in response to CO2 concentrations that have already occurred in North America. All right, take-home messages. Aspen secondary chemistry is highly variable in response to genetic, environmental, and developmental stage. Secondary chemistry is important in defense against insect and mammalian herbivores, but chemical defenses are not effective when herbivore pressure is high. Strong trade-offs exist between growth and defense in aspen genotypes, and aspen chemistry influences ecosystem functions such as nutrient cycling. And finally, aspen chemistry contributes to a mosaic of diverse community interactions and ecosystem functions at the landscape level. And with that, thanks to my many graduate students, postdocs, who've all contributed to this work, to my funding sources, NSF, USDA, and the Department of Energy. And one final unabashed advertisement: got chemistry, need a collaborator, contact us. We'd be glad to help out. And I'd be glad to take some questions. Yeah, Paul.
Just on the very last point there, you talked about the face sites and trade-offs between CO2 and ozone. In my previous work, we did a lot of surveying for ozone damage across the whole country. It was purported that ozone damaged aspen. There's a lot of evidence for that in the upper Midwest and East, but I haven't seen very much work that we can relate to that in the West and East. It seems a little confused or not, the question is not answered as to what those effects are. So we look forward to any kind of systematic survey or some sort of ozone damage to aspen, because this could be a significant factor in the health of aspen on a regional basis.
Right, yeah. Surprising. I'm not that familiar with ozone work in aspen out west, and obviously there hasn't been much done. It is so pronounced at the aspen face site that you can walk from one ring to another and see from 200 feet away what the treatment is. You don't have to look at the sign posted on the shed by each ring. There is a really dramatic suppressing effect of ozone on aspen growth. You get foliar symptoms? Yes, you get foliar symptoms. You get this stippling on the leaves, so there's this black stippling of the leaves, and then you typically get early leaf drop. Usually by mid-August, the leaves are starting to show some chlorosis, and by late August, they're dropping. Whereas in the ambient stands, they'll hold on to mid-September, and the elevated CO2 stands will hold on to the end of September. So we do get definite leaf signs of damage and definite stunting. And there have been studies with ozone across an ozone gradient from very small in northwestern Wisconsin to high in southeastern Wisconsin, where you've got these common gardens planted, and they'll document the same there. But I think it would, be easy to do it in the west by establishing some common gardens in high and low ozone areas and seeing what happens. Yeah, Jeff.
Kind of a 2x2 matrix question for you. Can you characterize the defensive chemistry of, say, aspen versus conifers, and then on the other hand, the detoxification chemistry of bark beetles versus herbivores?
Yes. Okay, so the conifers, their defensive chemistry is dominated by terpenoid compounds. I'm not telling you anything you don't know. But especially, these monoterpenes, diterpenes, and sesquiterpenes. Aspen does not produce them in any significant amount whatsoever. I mean, they'll release some volatiles that are terpenoid in nature. They do not have the resinous diterpene stuff that conifers have. right? So the chemistries are very different. Conifers do produce some tannins, but they're structurally different from the tannins produced by poplars. So the chemistries of the two groups are very different. Insects that feed on poplars degrade, detoxify the phenolic glycosides via an esterase-based system. Those phenolic compounds, these phenolic glycosides, are all esters if you look closely at the structure. And if you have high gut levels of esterases, you can break them apart and survive by feeding on it. If you inhibit esterase activity by feeding them an esterase inhibitor, the toxicity goes way up. That's different from insects that feed on conifers that are relying primarily on P450 enzymes to break down the terpenes. So again, there are different metabolic pathways that the insects are employing to defend against the two different groups of secondary metabolites.
And sort of carry this over in the previous talk with the emergence of what have been secondary bark fields in Aspen. You would hypothesize that they were the Aspen report very fully defended at this time.
And I was really interested to hear that. As far as I know, nobody's done any work with aspen bark beetles in relation to feeding to genetic variants or anything. You might know more about that, Jim, but that's a whole new world that's opened up to me. Yeah.
Let's make this one last question. Sam?
Rick, the base data looked like the divergence in the treatment effects occurred in a single year, 2000 and 2001. The last slide you showed. What was going on in terms of climate that year?
I'd have to go back and think. We've had very dry years, we've had very wet years, and it's true that the treatment effects are more pronounced in certain climatic conditions than others. I suspect the treatment effects are more pronounced in dry years than wet years because trees with high CO2 function better under droughty conditions than under low CO2. That's probably what was happening. I have to go back and look.
Thank you.